Hello, my name is Mike Woerdemann from the new company Evident, and I'm product manager for the Olympus high content screening system ScanR and the Olympus deep learning solution TruAI. Let me share my screen. Today I'm going to talk about TruAI in high content screening and automated microscopy analysis. First, let me introduce the concept of high content screening in the words of a microscopist. So high content screening means that we typically have a larger sample, like a well plate depicted here. A well plate consists, for example, of 96 wells. Each well needs quite a few images to acquire the whole area of the well. This means we need highly automated microscopy to acquire the whole plate, or maybe multiple plates in a systematic and reproducible way. This needs to be very robust. And what we typically end up with, what we want to end up with, is a large amount of image data. This can be a few images, like 10 images or 20 images, but it can also be something like 10,000 or even more images for a whole well plate. This means we can and have large statistics. So for each well, we have, for example, 100 images, and each image is a microscope image with, for example, 20x magnification. So we've also high content of information. So we see details of each cell, of each object. And the challenge now is to extract the relevant information from this large heap of images. And this means we need some kind of automated image analysis. So getting objects from the images. And we need some tool to explore the image content, to really get a hook on the really interesting features, the biologically interesting features. And we need some kind of automated data analysis. So get really the results, the final results from the analyzed images. So in the end, what we need is some kind of analysis protocol which describes the whole way from the raw images to the final results. And within our software, within the Olympus ScanR software, we call this an assay. So an assay is the analysis protocol. I will need that later. The foundation of all analysis is a good segmentation of all instances within the image. So with instances, I mean the nuclei, the cells, it might be cell clusters, it might be spots, it might be organelles within cells. So we need a good segmentation. And from this good segmentation, we can draw all the results and draw the conclusions. TruAI, which is Olympus deep learning solution, can deliver highly accurate results for the segmentation. There are many ways to do that. We offer tools to train customized networks to really go into a specific application and analyze images that are really difficult to analyze. But today I want to focus on the other side, the standard applications. So it is quite standard to have the aim to find all nuclei within a sample, or finding all cells, contouring and detecting all cells. So the TruAI in the latest version comes with pre-trained networks for nuclei, cells, and spots. And pre-trained means that you can open the network, run it, and it will deliver the results without any customization, without any training, without any need to do something. It will just work out of the box. And this is what I'm going to show in the live demonstration. Let me switch the share. Going to the software. Okay, this is a ScanR analysis main interface. So I'm focusing today on the analysis part. So we assume here that we have acquired the images already, and we are now aiming at understanding the images and analyzing them to get our results. I would like to open a very standard scan at the beginning. So what I do here is I open a scan. A scan is a set of images from one acquisition. So a scan can be well plate, for example. It has typically multiple images. So in this case, it only has approximately 20 images, but it can easily be hundreds of thousands of images. Maybe I visualize this in green here. It's a little easier to see. What I see here in the main UI is an example image of the scan. I can look at the more examples. I see the nuclei of a 10x scan of a U2OS cell line, and I see the nuclei state in DAPI. What I will do now, I will open an assay, an analyze protocol, load assay here, and I say I want the nuclei assay standard. Standard means this is not yet TruAI, so no deep learning involved here. And before I started, what I need to do is I need to adapt the settings, because the properties of the image, they depend on the magnification, on the brightness of the dye, of the skills of the experimental preparator. It depends on many external parameters, which may change from scan to scan, from sample to sample. And I need to check here. So here's the threshold. Everything above this threshold will be considered one nucleus. I need to decide if I want to split potential double detection. So if I switch this off, for example, I see here that these two nuclei here are wrongly detected as one object. I could change the threshold, of course, but there is always a sweet spot I need to find to have the best possible settings here. So this requires some experience, and with my experience, I set these settings. I can validate them with other example images. Seems to work decent, and I say okay, and I run this assay. I run the protocol on the whole data set. So what I will see soon here is data showing up here. So what we see here is well known to the people who know flow cytometry. Let me still explain briefly. So I have in all these plots two freely selectable axes. So on the top right, what I selected is the area of the objects that I detected on the x-axis, and the mean intensity of the fluorescence on the y-axis. So now each dot here represents one nucleus in my example. So if I click here on any of the dots, what I will see here that the corresponding nucleus is detected, is visualized in the main UI. So there are a few thousand here, and I can easily have a look at the bright ones, the dark ones, the small ones, and the big ones. So what I see here is two clusters in the plot on the top right. And clusters are typically something interesting. What I see here in particular is a cell cycle. So this is a fixed cell sample. So why can I see a cell cycle visualization? Because there are so many cells that I acquired that I find representatives in any state of the cell cycle. Let me start here somewhere with the nuclei, which are relatively small or have medium size and are quite dim. And if I go along these clusters, I see that the nuclei are increasing in size. So more concretely, the DNA staining is increasing in size. So I see here replication of more DNA. Then I go along this cluster here, I see the DNA condensing, getting smaller and smaller and more intense and more intense. At the very top, I see the really condensed nuclei with the full DNA content, with the full intensity. And if I go to the left here, then what I see is the nuclei directly after splitting. And this might be my question here, how many the fraction of nuclei that just undergone mitosis in relation to the nuclei which are in the G2 states. So let me see here if I pick them properly. So these are the nuclei just divided, and I can easily measure them. So the measurement is based on what I show here. I show the segmentation and the exploration of the data. The measurement is the not so interesting step, which is more the routine work afterwards. It can also be defined in the assay, but I don't want to show that here. But I also see, in addition to the two clusters here, is objects outside the clusters. These are, for example, the very big objects here. These are typically double detections, like this one here, and some nuclei which are have a strange shape for whatever reason of preparation or biological reasons. And I see very small ones. They're typically false detections because the detection is not perfect. But in general, this does not matter too much because I have so many objects which are valid that this is well below 1%, maybe even much less of false detections. So I can do a decent analysis here. But I needed to adapt it carefully to the data. And if I would work on the next scan, I would have to readapt. So let's try another approach. I now open a TruAI assay. So I open here the nuclei detector with TruAI, which has defined as the most fundamental step of segmentation to use a pre-trained network. It uses a pre-trained nuclei network. If I open this and press run, it will start immediately of analyzing the data. It takes a few seconds to load the images from disk here. After a few seconds, I see the data points coming up here. They are now detected with the neural network. What I see here is that the clusters are better defined, which is a hint to better segmentation. So I see that really the nuclei are well detected here. I can see, of course, the same that I saw before, analyze the same that I analyzed before. I can here have a look at the nuclei directly after mitosis. And what I see here in contrast that the fraction of bad detections, of wrong detection is really low. So everything that is here with unusual size is really something that is either not a valid nucleus or some nucleus which has changed due to some treatment or something. But this is a really low fraction. So 99.9% of the detections are really valid nuclei and can be used to do the analysis of the biology inside. So this worked out of the box without any adjustments. So let's see what happens if I open another scan without changing the analysis protocol. So I go now to the next scan here that I have prepared. Let's see, it's 4x, so different magnification, different cell lines. This is HeLa cells, DAPI staining, different size of the nuclei, of course. And I'm not sure if it's visible in the stream here, but there's also different background. So there's a noisy background here. So this is a completely different situation. So I would have to adapt the classical assay, but with the TruAI pre-trained networks, I just press the run key. And what I expect now is a perfect segmentation of these images, so multiple images in the scan, not only one image, but I guess again here 20 images for the example. And what I see here coming up soon is a similar structure, again, the cell cycle. So these stripes here, because the cells, the nuclei are so small, so that they're like a discrete number of pixels, but they are perfectly detected. I can here go through all the nuclei, almost no wrong detections here, just a very, very few, and I can exclude them easily. And let's do the same again that we did before. Look at the nuclei that just have undergone mitosis. I see them here, just a few representatives. There are much more in the sample, but it's easy to find them. And of course, then it's also easy to quantify them. Okay, going to the next scan. So we had now 10x U2OS cells, the nuclei of these cells, DAPI HeLa cells. Now I go to another cell line with 20x magnification. So again, different magnification. And what I have here is quite a challenging situation. So it's somehow not a monolayer, so cells overlapping a little bit. I have some out of focus cells. So this is really, really challenging. So let's see what the pre-trained networks with the same assay I used on all the scans before does. So what it basically does is a very decent job. So I can see here that the nuclei detected well, even strangely shaped nuclei in some kind of meta state are visible here. Let's go to something where it's very difficult. So here there is no obvious solution because it's overlap. Let's see what the network detected here. So basically excluded them, that makes sense. And let's go somewhere else, maybe here. So these networks are, I think this is human level, maybe not the super expert of the application level, but for pre-trained networks, this is really something that we can work with. And if I look here on the nuclei that just have undergone mitosis, or maybe shift this a bit, I think I picked too many, maybe here, and I see really the nuclei which are quite condensed and can be measured. Okay, let's pick maybe a more sophisticated example. Here I pick a scan where no nuclear marker is or has been acquired at all. So this is just a tubulin marker in HeLa cells with 10x magnification. So there is no nucleus marker. So with classical means, it's quite challenging to find nuclei here, right? So I would have to do much more than just thresholding. I would need to either invert or do some fancy investigation, which is always customized to this specific application. But what I do here, I just use the standard assay with the standard pre-trained network for cell detection. And what I see here, it does a decent job, even though this is not a nucleus channel. So it will find almost all the nuclei. It's not perfect. There are errors, definitely. So this is not easy to solve. Like here, for example, if for a human, it would also be quite challenging to find the nuclei correctly. But the pre-trained network do a very good job. I think I can show the overlay here to get an idea. So there are errors here, 1, 2, 3, maybe 1% of errors, but I find 99% of the nuclei and don't only find them so that I can count them. I also find a good prediction of the contours. I can measure now, for example, the FITC signal within the nucleus shape or outside the nucleus shape. I can do ratio calculations. I can do quite a lot here. But this obviously is not a scan which was acquired to detect nuclei. Here I see basically something that I would maybe want to use to detect the cell shape. So let's load another assay, another protocol to analyze. So far I use the nuclei TruAI, now I use the cells TruAI. I open that, run it. And now the same data, different pre-trained network. So the second one, the cell one. And what happens here is, I mean, this is a difficult job for classical image analysis, and there will be no perfect solution. So there is no obvious perfect contour, but what the pre-trained networks deliver here is very close to a perfect contour, I would say. So we can have a look at some representatives of all states here. So I can see here that, for example, even in this very noisy part of the sample where probably we had a lot of bleaching due to multiple acquisitions, it finds a very good estimate of the cell shape. I mean, this is not 100% perfect, but I think it's 99% perfect. And this is a very good foundation of a good analysis. Let's see if we can find more interesting examples even. I go to the COS7 cells where we have a 20x magnification and where the mitochondria are stained. So this is maybe not what you would acquire to detect cell shapes, but let's run the network here and see what happens. And what it does is it gives you a good prediction of the cell shape. So again, here it's difficult to say what the perfect cell shape is because the mitochondria are fragmented, obviously. But this is something we can build analysis on. And it's always reproducible. It will do the same in the next sample. It will have no bias like a human labeling person might have different rules that it applies from sample to sample. Here, this is quite reproducible. And this is really a very good cell segmentation giving the challenging situation here. Let's look at another example. This one here is, I picked this one because it's a really bad image quality. So there were reasons to acquire it with this low signal. But in the end, if we have now the task to segment the cells here, it's a cell line which I don't know. But from the image analysis point of view, it's really challenging. And it does not do a perfect job here. So there is no perfect solution to this, but does a decent job. It will like give the best probability or the best prediction. Here, this one is not a good one, but I would say here at least 90–95% of these excuse me, cells are well detected. This at least gives me the chance to do good statistics, to do density measurements of cells within different wells, for example. Let's switch to the next one, have one cells. Again in 20x magnification. I think here I even have a nucleus marker, which is good for us, for the human eye, to have a better reference here. Let's run the cell detection network here. Wait a few seconds, and right, here it comes. So what it does here is also amazing, at least for me, who has spent a lot of time to analyze this kind of structures with classical means. So the pre-trained neural network gives here a really good prediction of cell shapes. So for example here, okay, this is not good. This is a double detection. I would say this is too challenging to expect a perfect result. But I think this is like a 95% result, and we can do a lot of statistics, like measuring the DNA content within the cell envelope, or again counting, doing geometrical measurements like circumference or diameter or something. So here, a lot of nicely detected cells. Okay, let me finish or finalize this demonstration with something I want to show, because so far I only showed segmentation on one channel. Of course, this also works on multiple channels. I will rerun this here to give you an impression of the time it takes. There are only a couple of images. Let it be like 10–15 images, just demo set. I run it. What I have here is in blue. It's purple because of the overlay. In blue, the nucleus. In red, the mitochondria. And in green, some kind of cell marker. And what I have here is already finished. What I have here is a really good segmentation of the cells. Visualize differently of the cells and the nuclei. Okay, and with this I would like to finish the presentation of the software side and finish this workshop with a short glimpse on other examples to show a little bit more the broadness where the pre-trained networks can be applied. Okay, just a selection of examples. So these are stem cells where the nuclei are marked with DAPI in a 20x wide field scan. And what we see here is that the segmentation is close to perfect. We see here red 1 cells with a cytosolic staining in 10x wide field. Again, the segmentation of the cells is very good. Interesting structure, kidney organoid cells, where the images have been acquired with a 30x spinning disk confocal. So we see a strong optical sectioning here, and the pre-trained networks find all the nuclei within the focus layer. Another cell line with a tubulin staining in 20x wide field. Here we also get a decent segmentation of the cells with very little errors, if any. We have BPAE cells with an actin staining acquired with 40x confocal microscope, and we see also a plausible and good segmentation. HeLa cells with DAPI staining, where we have used the 40x spinning disk confocal, we see a strong optical sectioning here, all the structures within the nuclei, also possible to segment and analyze with the pre-trained networks easily. Something completely exotic, a muscle fiber. Muscle fibers shown here, the cell borders are marked in 20x wide field, and the pre-trained networks give a very good representation of the cell contours. Human tissue cells with DAPI staining of the nuclei in 20x wide field. Here there is no perfect solution because the cells are overlapping and they are like shadowing each other. But what the pre-trained network suggests here is certainly a good start for an automated analysis. HeLa cells with tubulin staining, we saw that example before, and now here is shown that the nuclei and the envelope of the cells are reliably detected. I think this is the last example, COS7 cells with the mitochondria staining now in 40x confocal acquisition. And also here we see the successful segmentation of the cells and the nuclei. And with this, I would like to finish this workshop. If you have any questions or requests, please drop me an email at mike.woerdemann@olympus.com, mike.woerdemann@olympus.com. And with this, I would like to finish and thank you very much for your attention.